All right, let's unpack this. Have you ever truly stopped to wonder what really happens to that delicious meal after you swallow it? We're talking beyond just the basic idea of digestion. There's a whole universe inside you, a molecular alchemy, intricate pathways, and vital processes that painstakingly turn food into us. It's a process so fundamental, yet so incredibly complex. Our mission today, you might say, is a deep dive into the fascinating world of nutrition, digestion, and absorption. We've pulled from a rich set of sources to give you, hopefully, a master-level understanding. And believe me, we're not just scratching the surface here. We're going to explore every single concept, right down to the unexpected quirks and the profound implications. We're aiming to equip you with the kind of insights that truly differentiate the well-informed from the simply aware. What's one aspect that consistently surprises you when delving into this field?
What's consistently fascinating is how fundamentally important these processes are, not just for basic survival, right, but for every aspect of optimal health. Think about it. A robust immune system, peak cognitive ability, even your emotional well-being, they all begin with how efficiently your body extracts and utilizes nutrients from your food. This isn't just a lesson in biology, it's truly the foundational understanding of well-being. It influences every single aspect of our lives, from daily energy levels to long-term disease prevention strategies. It really does touch everything.
That's exactly why we're going so deep today. Throughout this dive, you'll uncover the precise step-by-step mechanisms behind how your body meticulously utilizes everything from complex carbohydrates to, say, a tiny speck of iron. We'll delve into how it measures energy, how it stores it, and perhaps most crucially, what happens when these finely tuned systems encounter a glitch or, go awry. By the end of this, you'll gain insights that will genuinely change how you think about what you eat, moving beyond simple calories to a profound appreciation of your body's biological sophistication. You're essentially about to master the inner workings of your own fuel system. So let's begin at the very beginning. What does your body actually need from that plate of food? Beyond just water, there are 6 essential components that form the foundational pillars of our nutrition. We're talking about metabolic fuels, which encompass carbohydrates and lipids, then protein, fiber, minerals, vitamins, and a special category called essential fatty acids. But what exactly do each of these contribute? What are their roles?
Yeah, each one plays a distinct and frankly irreplaceable role. Think of metabolic fuels, your carbohydrates and lipids, as your body's primary energy currency. They're the immediate source of power for pretty much every cellular activity, whether you're thinking, lifting weights, or simply breathing.
Right, the basic fuel.
Exactly. Then there's protein. Now, protein is far more than just muscle builder fuel. It's absolutely crucial for the continuous growth, repair, and turnover of all tissue proteins throughout your body, from your skin to your internal organs. It can also serve as an emergency fuel source, but its primary function is really structural and functional.
Okay, structure and function. Makes sense.
Next, fiber, often called roughage. Right. It's unique because it's not digested for energy in the same way. Instead, it provides essential bulk in the intestinal lumen, which is vital for maintaining digestive health, promoting regular bowel movements, and very importantly, feeding your gut microbiome.
Feeding the good bacteria.
Precisely. Without it, your internal plumbing simply doesn't work as smoothly. Then you have minerals. These are the inorganic elements. They aren't just for bones, they have very specific metabolic functions, often acting as vital cofactors, essentially helper molecules for countless enzymes. They enable the biochemical reactions that keep you alive.
Like tiny keys for enzymes.
That's a great way to put it. And finally, vitamins and essential fatty acids. These are organic compounds needed in much smaller, almost microscopic amounts, but they are absolutely indispensable for a multitude of other metabolic and physiological functions, acting like those little keys again, unlocking crucial processes all over your body. It's an incredibly diverse and interconnected team.
It's truly incredible how the body takes these complex forms of food we eat and transforms them, because the bulk of what lands on your plate, things like large polysaccharides from starches, complex triacylglycerols from fats, and intricate proteins, they can't just be absorbed as they are. They're simply too big, right?
Exactly, too big and complex. They must first be painstakingly hydrolyzed or chemically broken down into their much simpler constituent units.
So polysaccharides become simple monosaccharides.
Right. And triacylglycerols become fatty acids, and proteins are reduced to individual amino acids. Only then, at that molecular level, can they be absorbed and truly utilized by your cells.
It's like breaking down a massive, intricate Lego structure into individual bricks before you can use them to build something entirely new. something that becomes you.
That's a perfect analogy, and that meticulous transformation isn't just reserved for the big macronutrients either. Even minerals and vitamins, though they're much smaller molecules, still need to be actively released from the complex food matrix they're embedded in.
So they're kind of logged in.
Often, yes. Think about how tightly some nutrients are bound within plant fibers, for instance. Your body has sophisticated enzyme-driven mechanisms to liberate them before they can even be presented for proper absorption and utilization. This isn't an automatic passive process. It's an active and selective event that requires specific conditions, specific enzymes, to work efficiently.
Without these mechanisms, many vital nutrients would just pass right through.
Essentially, yes, they'd be unavailable. Hashtag tagged Habibi, global nutritional realities, and their consequences.
Okay, so moving from those microscopic details of digestion to the broader global picture, it's impossible to ignore a stark and frankly troubling paradox in today's world. We have widespread undernutrition existing simultaneously with increasing overconsumption. It's a real tale of two extremes.
It really is. And when we look at undernutrition, the consequences are profound and systemic. They're particularly devastating in vulnerable populations. For instance, it directly leads to impaired growth, not just in physical stature, but also critically in cognitive development, especially in children. That has lifelong implications. It results in a severely defective immune system, making individuals far more susceptible to recurrent infections and chronic diseases, creating this vicious cycle of illness.
Right.
And on a societal level, it critically reduces work capacity, which perpetuates cycles of poverty and hinders economic development in affected regions. This isn't just an abstract problem, it's a very tangible, widespread global issue impacting millions of lives right now.
And the flip side, overconsumption, presents its own rapidly escalating global health crisis.
Yeah.
It's almost a mirror image in terms of imbalance. We're seeing alarming increases in rates of obesity, the widespread development of type 2 diabetes, a dramatic rise in cardiovascular diseases, and even strong links to certain types of cancers.
And it's spreading. While these issues were historically prevalent in developed countries, they are now rapidly rising in developing nations as dietary patterns shift globally. It's a crisis that spans continents, just from a different angle of nutritional imbalance.
It's not just about overall calories either, is it? There are specific nutrient issues too.
Exactly. Beyond these broad categories, specific micronutrient deficiencies remain major health concerns globally, often silently eroding public health. Vitamin A deficiency, for instance, is a leading cause of preventable blindness and severely impairs immunity. Iron deficiency, which is shockingly common worldwide, causes anemia, leading to chronic fatigue and reduced cognitive function and productivity. Iodine deficiency, another silent threat, can result in intellectual disabilities and goiter. These, along with deficiencies of other essential vitamins and minerals, are significant contributors to widespread ill health in many countries, undermining the very foundation of public health.
Now, it's true that in the developed world, overt, classic nutrient deficiency diseases like, say, scurvy or rickets are relatively rare these days.
Generally, yes.
But that doesn't mean everyone's getting optimal nutrition, right?
Yeah.
Vulnerable populations still exist, even in affluent countries.
Yeah.
And here's where the real insight lies, I think. A point that truly changes perspective. We often think, no deficiency, no problem. But the profound truth is, simply getting enough to avoid a diagnosable deficiency disease is worlds apart from getting optimal levels that truly power your immune system, sharpen your mind, and extend your healthy lifespan.
That's a critical distinction.
It's the difference between merely existing and truly flourishing, a distinction too often missed in our daily nutritional choices. This isn't just about survival. It's about thriving.
So we've outlined this incredible, finely tuned machinery.
Yeah, it sounds almost perfect.
Almost. But what happens when even one tiny gear in that system slips? Unfortunately, it's not always flawless.
Right. Let's maybe delve into some common digestive disorders. These really underscore just how delicate this biological balance is. They reveal the profound impact that seemingly small malfunctions can have on our overall health and nutrient absorption.
It's a complex system, and even subtle deviations can have significant impacts. Take gastric ulcers, for instance. While they're often associated with excessive gastric acid secretion, the crucial understanding now is their very common link to a Helicobacter pylori bacterial infection.
Ah, the H. pylori connection.
Exactly. This bacterium specifically undermines the stomach lining's natural defenses, making it vulnerable to acid damage. Then there are gallstones, those small, solid masses that form in the gallbladder. They arise from surprisingly minor changes in the composition of bile, often leading to the crystallization of cholesterol. A subtle chemical imbalance with potentially painful consequences.
Ouch.
Then another significant issue involves the failure of exocrine pancreatic secretion. This is a condition seen, for example, in genetic diseases like cystic fibrosis.
Okay.
The pancreas produces those vital digestive enzymes we discussed earlier. When the secretion fails, it results in severe undernutrition because food simply isn't being broken down properly. This leads to a telltale symptom called steatorrhea,
Which is?
excessive fat in the feces, because fats cannot be properly digested and absorbed. It's a clear sign something's wrong with fat digestion.
And a very common one many people experience, thankfully maybe less severe but still impactful, is lactose intolerance.
Very common.
This isn't a severe disease in most cases, but it certainly causes discomfort. It's directly linked to a lactase deficiency, meaning your body either produces too little or no lactase, the enzyme needed to break down lactose, the sugar, in milk.
Right. And when lactose isn't digested, it remains in the gut lumen. There, bacterial fermentation produces lactic acid and gas.
Leading to bloating, discomfort, diarrhea.
Exactly. All those uncomfortable symptoms upon consuming dairy products.
And then there are allergic reactions to foods. That seems like a different category again.
It is, and it's a fascinating example of the immune system's intricate and sometimes misguided role in digestion. These occur when your body absorbs intact peptides, small protein fragments that are large enough to stimulate an antibody response. Your immune system essentially mistakes these otherwise harmless food components for threats.
It's just an immune overreaction.
Precisely. It initiates an allergic reaction. Celiac disease is a specific and widely recognized example of this, where individuals have a distinct immune-mediated reaction, an autoimmune response, specifically to wheat gluten. It's a clear case of the body's defenses turning against itself in response to food.
What these disorders really emphasize, then, is that they're not just isolated problems. Each one dramatically illustrates how a specific enzymatic failure or a transport protein malfunction or an immune system misfire
Yeah, at any point in the digestive chain.
can have profound and far-reaching systemic effects. These failures directly compromise nutrient absorption and, by extension, profoundly impact overall health and well-being.
Absolutely. It truly highlights just how critical each finely tuned step in the digestive process truly is to your body's entire function.
All right, let's zoom in now on carbohydrates, a primary fuel source for our bodies.
Yeah.
The digestion of carbohydrates is a remarkable process. It begins the liberation of what we call oligosaccharides, essentially short chains of sugars. These oligosaccharides are then further hydrolyzed, broken down even more into their simplest forms, free mono and disaccharides. And it's only these simpler units that are finally ready for absorption into your bloodstream, right?
Exactly. And this complex process begins with a crucial enzyme called amylase. You actually have salivary amylase, which surprisingly starts breaking down starches right there in your mouth as you chew.
Starts immediately.
It does. But the main workhorse is pancreatic amylase, which is secreted into your small intestine. These enzymes are incredibly precise, acting like tiny scissors. They specifically catalyze the random hydrolysis of the glycoside bonds within starch molecules.
Those specific bonds.
Yes. This precise enzymatic action yields a variety of smaller carbohydrate fragments. First, dextrins, which are short branch chains of glucose, then simple sugars like maltose, a disaccharide of 2 glucose units, maltodextrose, a trisaccharide, and even small branch dextrins that come from the natural branch points in the original starch molecules. It's a systematic deconstruction, ensuring all the parts are ready for the next stage.
Now, when we talk about carbohydrates, a term that often comes up is the glycemic index, or GI. Let's define it precisely.
Okay.
The glycemic index is essentially a measure of how quickly and how much a specific carbohydrate test dose increases your blood glucose levels, compared to the blood glucose response you get from an equivalent amount of pure glucose. It's a way of rating the sugar spike potential of different foods.
That's a good way to put it. And to give you some context for what that means, pure glucose itself, or monosaccharides like galactose, are assigned a GI of 1, often expressed as 100% because they are the reference point.
The benchmark.
Exactly. Similarly, disaccharides like lactose, maltose, isomaltose, and trehalose, which are very quickly broken down into these simple monosaccharides, also have a high glycemic index, causing a rapid blood sugar rise. On the other hand, fructose and sugar alcohols like sorbitol or xylitol are absorbed much more slowly by your body, and therefore they have a significantly lower glycemic index. Sucrose, common table sugar, also has a lower GI compared to pure glucose because it's a disaccharide composed of one glucose unit and one fructose unit, and that fructose component slows down the overall absorption.
What's truly fascinating, I think, is how variable the glycemic index of starch can be. It's not a fixed number at all.
Not at all. Depending on the specific type of starch, whether it's from potatoes, rice, or corn, and crucially, how it's prepared or cooked.
Cooking matters.
Immensely. Its GI can range from near 1 or 100% down to almost 0. This wide range is a direct result of varying hydrolysis rates, essentially how quickly that particular starch is broken down by your digestive enzymes.
Well, things like fiber.
Right. Non-starch polysaccharides, what we commonly know as dietary fiber, have a GI of 0 because they aren't digested in the small intestine at all and thus don't contribute to blood glucose directly.
And this variability brings us to a key health insight, doesn't it? The benefit of low GI foods.
Yes. Foods that have a low glycemic index are generally considered more beneficial for long-term health. The profound reason for this is that they cause far less dramatic fluctuations in insulin secretion. Instead of a rapid spike in blood glucose, followed by a large insulin release, and then a subsequent crash.
The sugar roller coaster.
Exactly. You get a more gradual, sustained release of glucose into the bloodstream. This leads to a more stable insulin response, which is crucial for metabolic health. So this really begs the question for our dietary recommendations. How can understanding the glycemic index better inform our food choices and guide public health advice? Should we be focusing more on the GI of foods rather than just total carbohydrate counts to promote true metabolic well-being?
That's a really important question to consider. Following on from carbohydrates, let's dive into some truly unsung heroes of our diet. Resistant starch and non-starch polysaccharides, often collectively known as fiber.
Ah, yes, fiber. Crucial stuff.
What makes them so special is that they are not digested in your small intestine at all, which, paradoxically, is precisely why they are so powerful.
Exactly. Because they escape digestion in the small intestine, they journey onward to your large intestine. And there, they provide essential substrates, basically. Food for the bacterial fermentation carried out by your incredible gut microbiome.
Feeding the trillions.
Pretty much. These bacteria in your large intestine go to work on these compounds, and the result of this fermentation is the production of incredibly beneficial compounds, primarily butyrate and other short-chain fatty acids. This is where the magic happens.
And these short-chain fatty acids are far more important than most people realize, aren't they?
Absolutely. They are a significant, crucial source of fuel for your intestinal enterocytes.
Those are the cells lining the gut.
Yes, the cells lining your gut. So these undigested carbohydrates are not just passing through, they're literally feeding and nourishing the very cells that keep your digestive system healthy, intact, and functioning optimally. It's a remarkable symbiotic relationship that underpins your gut health.
That's amazing. They fuel the barrier itself.
Precisely. And what's truly fascinating here, and often a surprising fact for many listeners, is that there's compelling evidence suggesting that butyrate also possesses significant anti-proliferative activity.
Meaning?
Meaning it can help inhibit the growth of abnormal cells, potentially offering a significant protective effect against colorectal cancer.
Wow. So fiber isn't just about regularity or feeling full?
Not at all. It's a direct active contributor to colon health and a powerful potential guardian against disease. That's a huge takeaway. #tag down D8. Disaccharidases are brush border enzymes.
Now, to get those oligosaccharides fully broken down into their absorbable simplest units, we need another set of incredibly specialized enzymes called disaccaridases.
Right, the final cleanup crew for sugars.
The main players here are maltase, the bifunctional enzyme sucrose isomaltase, lactase, and tralase. Quite a lineup.
It is, and these enzymes are strategically located right on the brush border of your intestinal mucosal cells.
The brush border.
Okay.
Can you describe that?
Imagine it as a dense, microscopic shag carpet made of microvilli. It dramatically increases the surface area for absorption inside your intestine.
Got it. Like a massive surface area.
Exactly. And their function is critically precise. They are the final step in carbohydrate digestion. They hydrolyze disaccharides like maltose or lactose into their constituent monosaccharides glucose, galactose, and fructose. It's this precise last-mile breakdown that allows these monosaccharides, along with any that were directly ingested, to be absorbed directly into the body.
So without these enzymes right there at the surface,
Many dietary sugars wouldn't make it into your bloodstream, leading to digestive issues. They're absolutely crucial for that final step.
let's delve into a very specific and common example of where a desaccharидase deficiency can cause noticeable issues. Lactose intolerance. We often think of it as an adult problem, but there's also congenital lactase deficiency, which sounds quite serious.
It is, though thankfully, quite rare. This is where an infant is born with a complete lack of lactase. It leads to severe lactose intolerance and a profound failure to thrive if fed on breast milk or standard infant formula. It's a critical early diagnosis for survival.
Wow, okay. But the more common one.
Indeed. The much more common scenario, affecting a vast proportion of the global population, is adult-onset lactose intolerance. In most mammals, and fascinatingly, in the majority of human beings globally, lactase activity naturally begins to decline dramatically after weaning. Usually it becomes almost completely lost by late adolescence.
So it's actually the norm for it to decrease.
Globally speaking, yes. When this happens, ingested lactose remains undigested in the intestinal lumen because the necessary enzyme isn't there to break it down. There, it becomes a readily available substrate for bacterial fermentation by the gut flora, producing a surge of lactic acid, short-chain fatty acids, and gases.
And that leads to the symptoms.
That bacterial activity then leads to the uncomfortable symptoms we associate with lactose intolerance: abdominal discomfort, bloating, and diarrhea, especially after consuming relatively large amounts of dairy products.
What's truly captivating about adult-onset lactose intolerance is its evolutionary aspect.
Yeah.
You mentioned it's rare globally for adults to retain lactase activity.
Yes. The persistence of lactase activity into adulthood, known as lactase persistence, is globally quite rare. But it's notably prevalent in two very specific population groups: people of North European origin and certain nomadic tribes of sub-Saharan Africa and Arabia.
Why those groups specifically?
This genetic adaptation is strongly believed to have arisen in populations that historically relied heavily on domesticated animals and their dairy products for sustenance well into adulthood. It likely gave them a significant survival advantage during times of scarcity.
Wow. So it's a remarkable example of human coevolution with our food sources.
Absolutely. And to give you a truly unique example of this precise enzymatic adaptation to diet, consider marine mammals. They secrete high-fat milk that contains absolutely no carbohydrates. Unsurprisingly, their pups naturally lack the enzyme lactase from birth, as there's simply no lactose in their diet to digest.
That makes perfect sense.
It perfectly illustrates how enzyme systems are finely tuned and exquisitely adapted to the specific dietary environments and needs of a species, right down to the precise components of a mother's milk. It's a beautiful testament to biochemical evolution and adaptation.
So once those carbohydrates have been meticulously broken down into their simplest forms, monosaccharides like glucose, galactose, and fructose, how do they actually get from your intestinal lumen into your bloodstream? It's not just a passive process, right? There are specific mechanisms.
That's right. There are two primary, highly efficient mechanisms for this absorption in the small intestine. The first, and incredibly important mechanism, is a sodium-dependent active transport process.
Active transport needs energy.
Yes. Glucose and galactose are primarily absorbed this way, utilizing a specific transporter protein called SGLT1, that's the sodium glucose cotransporter 1. Think of it like a specialized high-efficiency pump that can even pull sugar into the cell against its natural concentration gradient, making sure you get every last bit.
Wow, uphill transport.
Exactly. What's crucial here is that SGLT1 is coupled to the sodium-potassium pump on the other side of the cell, which provides the energy for this uphill battle. These sugars also compete with each other for absorption at this transporter, an important detail in certain dietary contexts.
Okay, so that's glucose and galactose. What about fructose?
The second mechanism is carrier-mediated diffusion. This is primarily how fructose and sugar alcohols like sorbitol or xylitol are absorbed. Unlike the active transport for glucose and galactose, this method only allows movement down their concentration gradient from higher concentration in the gut lumen to lower concentration inside the cell.
So no direct energy needed, but still needs help.
Correct. They don't require direct energy expenditure to move, but they still need a specific protein carrier to ferry them across the cell membrane.
And this difference in absorption mechanisms has a direct, tangible implication, doesn't it, for how your body handles these sugars?
It certainly does. Because fructose and sugar alcohols are absorbed more slowly than glucose and galactose, if you have a moderately high intake of them, say, from a large fruit juice or a sugar-free candy, some may remain unabsorbed in the intestinal lumen.
And then the bacteria get involved again.
Exactly. When this happens, they can serve as a prime substrate for bacterial fermentation, similar to what happens with lactose in intolerant individuals. This fermentation can then lead to osmotic diarrhea as the unabsorbed sugars draw water into the intestine, causing discomfort and bloating.
Right, that makes sense. So regardless of how they get in, how do they get out into the bloodstream?
Good question. Regardless of which mechanism gets them into the intestinal cells, all absorbed monosaccharides ultimately need to exit those intestinal cells and move into the capillaries that surround your gut. This final exit is facilitated by another specialized transporter, the GLUT2 facilitative transporter.
GLUT2, okay.
From the capillaries, they then enter the hepatic portal vein and are swiftly transported to the liver and other tissues for metabolism and energy production. It's a highly orchestrated and incredibly efficient process from the initial breakdown to the final cellular uptake.
Now let's shift gears to lipids, or fats. The major dietary lipids are triacylglycerols, which make up the bulk of fats we eat, and to a lesser extent, phospholipids. Here's a basic puzzle. Your body is mostly water, and fats, well, they famously don't mix with water.
The classic oil and water problem.
Exactly. So how on earth does our digestive system even begin to process something so stubbornly hydrophobic? It's like trying to get an oil slick through a watery pipe.
Yeah,
You need some serious biochemical alchemy.
you've hit on the core challenge. Because of their inherent hydrophobic nature, these lipids cannot simply dissolve in the watery environment of your gut lumen. They must first be painstakingly hydrolyzed or chemically broken down into smaller units, and then, crucially, emulsified into incredibly tiny droplets.
Emulsified, like detergent does with grease.
Precisely. We call these droplets micelles, and they're microscopic, only about 4 to 6 nanometers in diameter. This emulsification is absolutely essential before they can be effectively absorbed into your body. Think of it like that microscopic detergent breaking down a large grease stain into tiny manageable beads so they can finally pass through the watery sieve of your gut.
And it's not just the dietary fats themselves that rely on these micelles for absorption. This is a key point, right?
Absolutely key. The fat-soluble vitamins, that's vitamins A, D, E, and K, along with other vital lipids like cholesterol and various carotenes, are all absorbed by being dissolved within these lipid micelles.
So the micelles act like tiny transport capsules.
Exactly. They package these essential but water-insoluble compounds for safe passage through your digestive tract.
This leads to an important caution then, a truly practical insight for daily nutrition.
Yes.
If you are on a very low-fat diet, particularly one that severely restricts fat intake, the absorption of carotenes and these crucial fat-soluble vitamins can be significantly impaired.
That's right. It raises a critical question that often goes unasked. Why do some well-intentioned low-fat diets, despite their intentions, sometimes miss the mark nutritionally? They might potentially create deficiencies in these fat-dependent vitamins.
So it highlights that fat isn't just a fuel source.
No, it's a necessary vehicle and facilitator for the absorption of other vital nutrients. It's all about balance, not just reduction.
So how do we begin breaking down these hydrophobic triacylglycerols? Where does it start?
Surprisingly, the process actually starts quite early. Triacylglycerol hydrolysis is initiated by lingual lipase, an enzyme secreted in your mouth, and gastric lipase, which acts in your stomach.
In the mouth and stomach. I thought fat digestion was mainly in the intestine.
The main part is, but it gets a head start. These enzymes begin to snip off one arm of the fat molecule, specifically the SN3 ester bond, forming 1-viryl-2 diacylglycerols and releasing a free fatty acid. And interestingly, these free fatty acids themselves actually start to act as mild emulsifying agents, beginning to disperse the fats even further.
Okay, so a bit of predigestion.
Exactly. But the real heavy lifting and primary digestion of fats happens in your small intestine, with the powerful enzyme pancreatic lipase. This enzyme is incredibly precise, like a surgical tool. It focuses on very specific parts of the fat molecule, the ester links at positions 1 and 3 on the triacylglycerol.
It's off the outer arms.
Precisely. Its job is to break off these arms of the fat, leaving behind a core body called a 2-monoacylglycerol and more free fatty acids as the major end products of this digestion. Crucially, pancreatic lipase requires another pancreatic protein called collapase for its full activity. Collapase acts like a chaperone, helping lipase bind to the fat droplet despite the presence of bile salts which could otherwise block it.
That's intricate. And what's truly intriguing is how this specific enzymatic action has found an unexpected application in medicine, right?
Yes, inhibitors of pancreatic lipase are actually used in the treatment of severe obesity.
How do they work?
By preventing this enzyme from doing its job breaking down and thus allowing the absorption of dietary fat, these inhibitors reduce calorie intake from fat, leading to weight loss. It's a direct therapeutic use stemming from understanding this precise biochemical pathway, essentially a fat absorption blocker.
Fascinating. But is all the fat completely broken down?
Not always 100% down to glycerol and fatty acids. While pancreatic lipase is highly effective, there's also pancreatic esterase in the intestinal lumen, which can hydrolyze monoacylglycerols, but they are relatively poor substrates for it. What this means in practical terms is that only about 25% of the ingested triacylglycerol is completely hydrolyzed all the way down to glycerol and free fatty acids before absorption.
Only 25%. What happens to the rest?
The vast majority, about 75%, are absorbed as 2-monoacylglycerols and free fatty acids. These are then efficiently reassembled back into triacylglycerols inside the intestinal cells. It's a highly efficient process, tailored for quick reuse, not necessarily complete demolition first.
When we talk about fat digestion and absorption, we absolutely cannot overlook the unsung heroes, bile salts.
Definitely not. They're critical players.
These compounds are formed in your liver and then secreted into your small intestine via bile. Their primary role is like a microscopic detergent, right?
Exactly. They permit the emulsification of lipid digestion products, those fatty acids and monoacylglycerols, into those incredibly tiny micelles we mentioned earlier. Without them, your fats would just glob together, inaccessible.
And these micelles aren't just for the broken down dietary fats. Their composition is quite diverse.
Oh yes, quite sophisticated. They also incorporate dietary phospholipids and cholesterol. It's worth noting that your body actually secretes a significant amount of cholesterol, about 2 grams per day in the bile, in addition to the roughly half a gram per day of dietary cholesterol you might consume.
Wow, so we secrete more cholesterol than we typically eat.
Often, yes. So these micelles are a complex blend of both ingested and endogenously produced lipids, forming a highly effective transport vehicle for all of them.
What's absolutely crucial about micelle function is their unique combination of size and solubility, isn't it?
Yes, that's key. Being less than 1 micrometer in diameter and completely soluble in the watery environment of the intestinal lumen, they perform an invaluable service. They allow the lipid digestion products, including those vital fat-soluble vitamins, to traverse the watery gut environment and come into close, intimate contact with the brush border of your mucosal cells.
Right up to the absorption surface.
Exactly. This close proximity is essential for efficient uptake into the cells. Without micelles, these hydrophobic nutrients would essentially just clump together and pass through your system unabsorbed. A truly wasted meal, nutritionally speaking.
It's amazing efficiency. And the bile salts themselves, do they get used up?
Well, here's another testament to the body's incredible efficiency. Most of these bile salts don't just disappear after their job is done. They remain largely within the intestinal lumen and are then actively reabsorbed from the ileum, the final section of your small intestine, back into what's called the enterohepatic circulation.
Meaning they get recycled.
Yes. They're recycled back to the liver to be reused again and again for fat digestion. This remarkable metabolic economy minimizes waste and ensures a continuous supply for efficient fat processing.
Now, once those precious fatty acids and monoacylglycerols make it inside the intestinal epithelial cells, the lining of your gut, the story isn't over, is it? A crucial transformation begins.
That's right, it's reconstruction time. Specifically, any 1 monoacylglycerols that get absorbed are hydrolyzed into their constituent fatty acids and glycerol. But here's where it gets really clever. The vast majority, the 2 monoacylglycerols, are predominantly reacylated back into their original form, triacylglycerols.
Put back together.
Yes, via what's known as the monoacylglycerol pathway. It's essentially an internal reassembly line, putting the fat molecules back together inside your cells.
And the glycerol, what happens to that?
Its fate is quite distinct depending on its origin. Any glycerol that was released in the intestinal lumen itself is absorbed directly into the hepatic portal vein, heading straight to the liver for further metabolism. However, glycerol that is released within the epithelial cells themselves is shrewdly reused for new triacylglycerol synthesis, but this time via a different route, the phosphatidic acid pathway.
So the body efficiently recycles what it can internally.
Smart.
Maximizing its resources, yes. From there, the newly reesterified long-chain fatty acids are incorporated back into triacylglycerol in the mucosal cells. But these reconstructed fat molecules are still too large and hydrophobic to travel freely in the watery bloodstream, right?
Correct. They need packaging. They're packaged into tiny, specialized transport vehicles called chylomicrons.
Ah, chylomicrons. I've heard of those.
They're like little fat delivery trucks, but they're too large to enter the blood capillaries directly around the gut.
So how do they get out?
They are secreted into the lymphatic vessels, which eventually merge and enter the bloodstream via the thoracic duct, a major lymphatic vessel located near your neck.
So it's an indirect route into the blood.
It is a specialized indirect transport system that ensures these fats eventually reach every cell in your body via the bloodstream.
What about shorter fats? Do they follow the same path?
Ah, good point. What's important to contrast here is the journey of short and medium chain fatty acids. Unlike their long chain counterparts, these are mainly absorbed directly into the hepatic portal vein as free fatty acids.
Straight to the liver.
Yes. This bypasses the need for chylomicron formation and lymphatic transport, simplifying their journey and allowing for more rapid delivery to the liver for immediate metabolism. It highlights the body's nuanced and efficient approach to handling different types of fats depending on their molecular size.
Let's talk about cholesterol absorption specifically. It's absorbed, as we mentioned, dissolved within those lipid micelles. And once it's inside the intestinal mucosa, it's mostly esterified, meaning a fatty acid is added to it before being packaged into those chylomicrons for transport out to the body.
Correct. Now, here's where it gets really interesting, especially for its profound health implications. And it's a crucial insight for anyone thinking about managing their cholesterol. We introduce plant sterols and stanols.
Okay, found in plants.
Yes, found naturally in plants. They have a very similar chemical structure to cholesterol, but with a slight difference, the B ring structure is saturated in stanols. What they do is compete fiercely with cholesterol for that esterification process within the intestinal cells.
They compete for the same enzyme.
Essentially, yes. However, plant sterols and stanols themselves are very poor substrates for this esterification, meaning they don't get esterified efficiently and tend to get kicked back out of the cell.
Okay, so what's the implication of that competition?
The fascinating implication is that it leads to an increased amount of unesterified cholesterol remaining inside the mucosal cells. It's essentially stuck there because the plant sterols are occupying the machinery that would otherwise esterify and package it.
Like a traffic jam at the cholesterol exit ramp.
Exactly. And here's the truly ingenious transport mechanism the body employs to deal with this stuck cholesterol. This unesterified cholesterol, along with other sterols like the plant ones, is actively transported out of the mucosal cells and sent right back into the intestinal lumen.
Pumped back out.
Yes, this is a critical point.
Yeah.
What this means in practice is that plant sterols and stanols effectively inhibit the absorption of both the cholesterol you consume in your diet and, crucially, the much larger amount of cholesterol that your body secretes into bile every single day.
Wow, it blocks both dietary and biliary cholesterol absorption.
Correct. The net result of this dual inhibition is a lower whole body cholesterol content, which in turn leads to a lower plasma cholesterol concentration. This is a remarkable natural mechanism for dietary cholesterol management, offering a powerful way to influence your body's cholesterol levels just by including these compounds in your diet. It's really quite clever.
Moving on to proteins, the body's essential building blocks for virtually everything.
Yeah.
Muscle, enzymes, hormones.
Absolutely fundamental.
One of the key challenges with digesting proteins, though, is that their native, intricately folded three-dimensional structure, their native conformation, makes them quite resistant to proteolytic enzymes, doesn't it?
It does. In that folded state, only a few peptide bonds are actually accessible for those enzymes to start breaking them down. They're tucked away inside.
So how does the body get around that?
A crucial first step, a non-negotiable prerequisite for efficient protein digestion, is prior denaturation.
Denaturation, unfolding them.
Exactly. Unfolding that complex 3D structure, making the internal peptide bonds accessible. This can happen in two primary ways. First, through the application of heat during cooking, which physically unravels the protein structure, making it much easier to digest.
So cooking helps digestion directly.
Significantly. Second, and crucially, through the powerful action of gastric acid in your stomach, which chemically denatures them. This explains not only why we cook our food, but also why your stomach acid is so incredibly potent. It's literally preparing the proteins for efficient breakdown by the enzymes that follow.
Okay, once proteins are denatured and unfolded, a whole powerhouse of enzymes, collectively called proteases, gets to work.
The protein demolition crew.
And these are broadly categorized into two main classes, right? Each with different specificities for which peptide bonds they target, ensuring a thorough breakdown.
Indeed. The first class is endopeptidases. These enzymes act like internal molecular scissors.
Cutting from the inside.
Yes, hydrolyzing peptide bonds throughout the protein molecule, specifically targeting bonds between certain amino acids. They are the initial actors in protein digestion. Their role is to take a large, intact protein and yield a greater number of smaller fragments, essentially chopping the long protein chains into more manageable pieces.
Can you give some examples?
Sure. Pepsin in your gastric juice is an endopeptidase. It specializes in catalyzing the hydrolysis of peptide bonds next to amino acids with bulky side chains, like aromatic branched chain amino acids and methionine. Then, in the small intestine, you have a powerful trio of endopeptidases secreted by the pancreas: trypsin, chymotrypsin, and elastase.
The pancreatic protease.
Exactly. Trypsin specifically targets and cuts peptide bonds adjacent to the amino acids lysine and arginine. Chymotrypsin, on the other hand, prefers bonds next to aromatic amino acids. And elastase works on bonds near small, neutral, aliphatic amino acids.
So they each have their favorite spots to cut.
Precisely. This coordinated attack ensures thorough fragmentation of the proteins into smaller and smaller peptides.
Okay, that's endopeptidases. What's the other class?
The second major class is exopeptidases. Unlike endopeptidases, these work like trimmers.
Trimming from the ends.
Yes, catalyzing the hydrolysis of peptide bonds one at a time from the very ends of peptides. Examples include carboxypeptidases, which are secreted in pancreatic juice and meticulously release amino acids from the free carboxyl terminal end of a peptide. Then there are amylopeptidases, secreted by the intestinal mucosal cells, which release amino acids from the amino terminal end.
So they attack from both ends.
They do. And finally, you have dipeptidases and tripeptidases located right on the brush border of intestinal mucosal cells. These are vital because they specifically catalyze the hydrolysis of di- and tripeptides, which are often too small to be substrates for the larger amino and carboxypeptidases.
They handle the really small pieces.
Exactly. So it's a multi-stage, incredibly precise demolition, ensuring proteins are fully broken down into absorbable units.
Now, here's a truly ingenious safeguard built into your digestive system. We've just talked about all these powerful proteases.
Very powerful.
Imagine if they were active all the time, everywhere. They'd quickly digest your own tissues. That sounds bad.
Very bad. So your body has a clever mechanism. Proteases are secreted as inactive precursors called zymogens.
Zymogens, inactive forms.
Yes. Their active site, the part of the enzyme that actually does the work, is masked by a small region of the peptide chain, rendering them harmless until needed.
So how do they get activated only when needed?
It's an intricate cascade of activation, ensuring these enzymes are only active precisely when and where they're needed, preventing self-digestion. For instance, pepsinogen, the zymogen for pepsin, is activated to its active form, pepsin, by the highly acidic environment of gastric acid in your stomach. And fascinatingly, once a little pepsin is active, it can also activate more pepsinogen, a process called autocatalysis.
It kickstarts itself.
Essentially, yes. It amplifies the activation process.
And in the small intestine, is it similar?
In the small intestine, this cascade continues with even greater intricacy. Trypsinogen, the precursor of trypsin, is activated by a key enzyme called enteropeptidase, which is secreted by the duodenal epithelial cells right at the start of the small intestine.
Enteropeptidase, the master switch.
You could call it that. Once trypsin is activated, it becomes the master activator for all the other major pancreatic zymogens. Activated trypsin activates chymotrypsinogen to chymotrypsin, proelase to elastase, procarboxypeptidase to carboxypeptidase, and proaminopeptidase to amidopeptidase.
Wow, one activates all the others.
It's an intricate sequential activation that ensures precise control. It prevents premature digestion of the pancreas itself and maximizes digestive efficiency exactly where it's needed in the small intestine. It's a beautifully choreographed biochemical dance.
So after this incredible orchestrated action of endopeptidases and exopeptidases, what you're left with are the final products ready for absorption.
Right, the building blocks are ready.
A meticulously prepared mixture of free amino acids, along with some diene to tripeptides, and even a few smaller oligopeptides. All of these are now in a form that the body can readily take in.
And their absorption mechanisms are quite specific and highly efficient. Free amino acids are absorbed across the intestinal mucosa primarily by sodium-dependent active transport.
Active transport, again, like glucose, needs energy and transporters.
Yes. And there isn't just one general transporter. Instead, there are several different amino acid transporters, each with specificity for the nature of the amino acid side chain, whether it's large or small, neutral, acidic, or basic.
Different transporters for different types of amino acids.
Correct. This specificity also means that amino acids carried by any one transporter will actually compete with each other for absorption.
Competition. Interesting. What about the small peptides?
As for dipeptides and tripeptides, they have their own unique and rather clever pathway. They enter the brush border of the intestinal mucosal cells intact.
They get absorbed as small peptides, not broken down first.
They enter the cell intact, yes. That's an important distinction. Once inside these cells, they are then rapidly hydrolyzed into free amino acids by intracellular peptidases. It's only after this final intracellular breakdown that these free amino acids are transported into the hepatic portal vein, destined for the liver.
So they enter as peptides, but leave the cell as amino acids.
Predominantly, yes. This two-stage absorption mechanism actually increases the overall efficiency of protein digestion and absorption. It provides another route besides just free amino acid transport.
Now here's a less common but incredibly significant absorption route that has profound implications for human health. While most proteins are meticulously broken down into individual amino acids or very small peptides, relatively large peptides may occasionally be absorbed intact, meaning they aren't fully broken down.
It does happen, yes. Usually in very small amounts, but it can happen. This can happen either by being taken directly into the mucosal epithelial cells, which is called transcellular absorption, or by actually passing between the epithelial cells, a route known as paracellular absorption, sometimes called leaky gut, though that term is often used loosely.
And this absorption of larger peptides is linked to allergies.
And this fascinating phenomenon is directly connected to a very common issue many people face: food allergies. Many of these intact peptides that are absorbed are large enough, with their specific three-dimensional shapes, to stimulate antibody formation in your immune system.
The immune system sees them as foreign.
Exactly. Your immune system recognizes them as foreign invaders, even though they're just food fragments. This stimulation of an antibody response is precisely the underlying basis of allergic reactions to foods. Your body essentially mounts an immune attack against something that should be a harmless nutrient.
Wow. So it raises an important question for us. Why is the body's sophisticated immune system sometimes overly reactive to otherwise harmless food components, leading to these often debilitating allergic responses?
It's a critical area of research. It truly highlights the delicate interplay between digestion, absorption, the gut barrier, and immunology.
Okay, shifting our focus now to the micronutrients, vitamins and minerals. We know they're essential for countless bodily functions, but how do they actually become available to our bodies from the food we eat?
Well, like the macronutrients, they are released from the complex food matrix during digestion. The digestive process breaks down the food structure around them.
But there's a catch, you mentioned.
Yes, there's a crucial caveat. This release is not always complete. And critically, their bioavailability, meaning how much of them is actually absorbed and utilized by your body, depends significantly on the type of food they're in.
So the food source matters a lot.
Immensely. And especially for minerals, bioavailability depends heavily on the presence of other compounds in the food known as chelating compounds.
Chelating compounds, what do?
These compounds have a strong tendency to bind to minerals in the intestinal lumen, forming insoluble complexes. This effectively makes the minerals unavailable for absorption, even if they've been physically released from the food matrix.
So they grab onto the minerals and prevent uptake.
Exactly. It's a complex chemical dance within the gut that determines whether your body can truly access these vital nutrients. Things like phytates and oxalates are common examples.
So once released, assuming they haven't been chelated, how do these tiny but mighty micronutrients get into our system? They're different categories, right?
Yes, there are two main categories of vitamin absorption, largely mirroring their solubility properties, which dictates their journey through the body.
Fat soluble and water soluble.
Precisely. The fat-soluble vitamins A, D, E, and K are absorbed in conjunction with dietary fats. They dissolve beautifully within those lipid micelles that we discussed earlier.
So their absorption depends on fat digestion working properly.
Absolutely. It highlights their direct dependence on proper fat digestion and absorption. If fat digestion is impaired, so too will be the absorption of these vital fat-soluble vitamins, leading to potential deficiencies.
Okay. And the water-soluble ones, like B vitamins and C?
In contrast, water-soluble vitamins and most mineral salts are absorbed from the small intestine by one of two primary methods. Either through active transport, which requires energy and specific transporter proteins to move them, often against a concentration gradient.
Uphill again?
Often, yes. Or by carrier-mediated diffusion, where they move down their concentration gradient, often with the help of a protein carrier. Once inside the cells, they are often bound to intracellular proteins to achieve a concentrated uptake, allowing the body to accumulate them efficiently.
And some have very specific needs, right? Like B12.
Exactly. Vitamin B12 is a classic example. It requires a very specific transport protein called intrinsic factor, secreted by your stomach for its absorption further down in the ileum. Without intrinsic factor, you simply cannot absorb B12 effectively, regardless of intake.
Let's focus on calcium for a moment. It's a mineral vital for bones, muscles, nerves, and countless cellular processes.
Absolutely essential.
And its absorption is uniquely and directly dependent on adequate vitamin D levels. You can have plenty of calcium in your diet, but if you're low on vitamin D, your body won't be able to absorb it effectively. It's like having a key but no lock, right?
That's a great analogy. Vitamin D plays a truly fascinating dual role in calcium absorption, acting both in the long and short term. First, it induces the synthesis of a protein called calbindin. This is an intracellular calcium-binding protein absolutely required for calcium to be absorbed efficiently across the intestinal cell.
So vitamin D helps build the absorption machinery.
Exactly, the long-term machinery. Second, and remarkably, vitamin D also acts much more rapidly. It triggers the swift recruitment of existing calcium transporters to the cell surface. This process is independent of new protein synthesis and allows for a rapid on-demand increase in calcium absorption.
Wow, both slow and fast action. Very sophisticated.
It is. However, several factors can significantly impair calcium absorption, even if vitamin D levels are perfectly sufficient.
Like those chelators you mentioned.
Precisely. For example, phytic acid, also known as inositol hexaphosphate, found abundantly in many cereals and legumes, has a strong tendency to bind calcium in the intestinal lumen, forming insoluble complexes and effectively preventing its absorption.
So whole grains could potentially block calcium?
It can be an issue, particularly with unleavened whole wheat products. Thankfully, yeast, used in leavened bread, contains an enzyme called phytase, which can dephosphorylate phytate and inactivate it, making calcium more available.
Yeah, so bread making helps.
It can. Another factor that can hinder calcium uptake is a high concentration of fatty acids in the intestinal lumen, which can occur if someone has impaired fat absorption, like in certain digestive disorders. These excess fatty acids can reduce calcium absorption by forming insoluble calcium salts, essentially soaps.
Okay. Anything else?
Yes, a high intake of oxalate found in foods like spinach, rhubarb, and certain nuts can also lead to calcium deficiency over time because calcium oxalate is also insoluble and thus unabsorbable.
So it's not just about what you eat, but about the complex interplay of nutrients within that meal? Bioavailability is key.
Absolutely. It's about the whole picture.
Now let's talk about iron, another mineral absolutely crucial for oxygen transport via hemoglobin and for numerous enzymatic reactions.
Critical, yes.
Despite iron deficiency being a remarkably common problem globally, its absorption is actually quite limited and critically very strictly controlled by your body. This seems like a paradox, doesn't it? Why limit the absorption of something so vital and often lacking?
It absolutely is a paradox, and the reason for the strict control is profound and deeply protective. Approximately 10% of the global population is genetically at risk of iron overload, a condition called hemochromatosis.
Iron overload.
Yeah.
That sounds dangerous.
It can be very damaging. Free iron is highly reactive and can generate harmful free radicals through non-enzymatic reactions, damaging cells and tissues. So, to mitigate the risk of these adverse effects, iron absorption is tightly regulated. It's a vital, ancient protective mechanism that prevents systemic damage from iron toxicity.
Okay, so how is it controlled? How does iron get in?
The transport of inorganic iron into the mucosal cell is a proton-linked process carried out by a specific protein called the divalent metal ion transporter, or DMT1. It brings iron in as the F2+ reduced state. Once inside, iron is carefully accumulated intracellularly by binding to a storage protein called ferritin, which acts like a safe deposit box within the cell.
Stored inside the gut cell. How does it get out into the body?
Iron leaves the mucosal cell via another critical transport protein called ferroportin. But here's the key regulatory step. It only exits if there is free transferrin in the plasma to bind to it.
Transferrin, the blood transporter for iron.
Yes, your body's main iron transport protein in the blood. If transferrin becomes saturated with iron, meaning the body has enough, any iron that is accumulated in the mucosal cells simply remains stored there in ferritin. Eventually, those intestinal cells are naturally shed into the gut lumen every few days, and the excess iron is lost with them, preventing systemic overload. It's a clever mucosal block.
Wow. And there's hormonal control too. Hepcidin.
Yes, this regulation is even more sophisticated. A small peptide hormone secreted by the liver called hepcidin plays a central master role in iron homeostasis. When your body's iron reserves are adequate or high, hepcidin is synthesized and acts to downregulate the ferroportin gene and possibly DMT1 as well, effectively limiting further iron absorption from the gut.
So it turns down absorption when stores are full.
Precisely. Conversely, in response to increased demand, such as hypoxia, low oxygen, anemia, low red blood cells, or hemorrhage, hepcidin synthesis is dramatically reduced. This reduction leads to increased ferroportin synthesis and consequently increased iron absorption from your diet. It's a beautifully tuned, highly responsive feedback loop that balances absorption with bodily needs.
So as a result of all this control, how much iron do we actually absorb?
As a direct result of this sophisticated mucosal barrier and regulatory system, only about 10% of dietary iron is typically absorbed from a mixed diet. And for many plant foods, that absorption rate can be even lower, just 1 to 5%, due to the presence of those chelating compounds like phytates.
Only 1 to 5% from plants. That's really low. Are there ways to enhance absorption?
Yes, certain factors can significantly enhance absorption. Inorganic iron, for instance, is absorbed in the Fe2+ reduced state, so the presence of reducing agents enhances its uptake. Vitamin C is the most effective in this regard. Even a modest intake of 25 to 50 milligrams per meal can significantly enhance non-heme iron absorption, especially when iron salts are used to treat anemia by keeping the iron in its more absorbable F2+ state.
So vitamin C with iron-rich meals is a good strategy.
Definitely. Alcohol and fructose also modestly enhance iron absorption, interestingly.
And what about iron from meat? Heme iron.
Ah, yes. Heme iron, which comes specifically from meat sources like red meat, poultry, and fish, is absorbed via a separate pathway, distinct from inorganic iron. It's considerably more available and efficiently absorbed by the body.
So meat is a better source in terms of bioavailability.
Generally, yes. But here's a crucial factor that often goes unnoticed, and it's a critical insight for optimizing your iron intake. The absorption of both inorganic and heme iron is impaired by calcium.
Calcium interferes with iron.
Yes. A simple glass of milk consumed with an iron-rich meal, for example, can significantly reduce iron availability from that meal.
Wow, that really complicates things. It raises an important question for you to ponder. How do we optimize iron intake given these complexities, especially for those who consume dairy regularly or are trying to maximize iron absorption through plant-based diets?
It requires a more nuanced, strategic approach than simply eating more iron-rich foods.
Yeah.
Timing and food combinations become really important.
Okay, let's zoom out again to the big picture of energy. Beyond the absolute necessity of water, the body's most fundamental and continuous requirement is from metabolic fuels.
Yes, the energy sources.
We're talking about fats, carbohydrates, and amino acids derived from proteins. These are the compounds that quite literally power every single function your body performs, from the blink of an eye to the beat of your heart.
Absolutely. And here's where the concept of energy balance becomes absolutely critical to your health. It's a fundamental equation. If food intake consistently exceeds energy expenditure if you're taking in more fuel than you're burning, the inevitable outcome is obesity, the storage of excess energy as fat. Conversely, if intake is consistently less than expenditure, it leads to severe emaciation and wasting, manifesting in severe forms like marasmus and kwashiorkor, which we'll delve into shortly. Two sides of the same coin of imbalance. Exactly. The profound insight here is that both extreme severe obesity and severe undernutrition are strongly associated with increased morbidity and mortality. Maintaining balance is key.
And to track this balance broadly, the body mass index, or BMI, is commonly used, right?
Yes, it's a common screening tool. It's calculated as your weight in kilograms divided by the square of your height in meters, kiloma, with a desirable range generally considered to be between 20 and 25, although its limitations are also recognized.
So how do we actually figure out precisely how much energy our body is expending? It's not as simple as guessing based on activity.
No, it requires careful measurement. There are a few sophisticated ways researchers and clinicians can determine your energy burn. The most direct method is called direct calorimetry. This involves literally measuring the heat output from the body in a sealed chamber.
Measuring heat directly.
Yes. It's conceptually straightforward. Heat is energy, but practically quite challenging and less common for routine use outside of specialized research labs.
Okay, so what's more common?
More commonly, energy expenditure is estimated indirectly from oxygen consumption. This highly precise technique is known as indirect calorimetry. It's based on a well-established metabolic principle. There's an approximate energy expenditure of 20 kilojoules, or roughly 4.8 kilocalories, for every liter of oxygen consumed.
And that holds true regardless of what fuel you're burning.
Remarkably, yes. This fairly consistent ratio holds true regardless of whether the primary fuel being metabolized is carbohydrate, fat, or protein, making it a very practical and accurate estimation tool in clinical and research settings using metabolic carts.
Interesting. And you can get even more detail, like what fuel is being used.
Yes. Building on that, the respiratory quotient, or RQ, provides even more granular detail about your body's fuel mix. The RQ is simply the ratio of the volume of carbon dioxide produced to the volume of oxygen consumed, VCO2, VO2. It's a powerful indicator of the exact mix of metabolic fuels your body is currently oxidizing.
How does that work?
For example, if you're burning purely carbohydrates, the chemistry dictates your RQ will be 1.0. If it's purely fat, it's lower, around 0.7. Protein is intermediate. So measuring the RQ of someone breathing into a metabolic cart gives you a real-time window into what fuels their body is utilizing.
Fascinating. Now, for something really cutting edge in measuring long-term energy expenditure outside of a lab setting, you mentioned dual isotopically labeled water.
Yes, the doubly labeled water technique, 2H2-18O. It sounds complex, but it's brilliant in its simplicity and accuracy for measuring total energy expenditure over days or weeks in free-living individuals.
How does that work?
It truly is ingenious, and this is where it gets really interesting for understanding energy expenditure in the real world. Here's the concept. When you ingest this special water, the deuterium 2H label, a heavy isotope of hydrogen, is lost from your body primarily in water through urine, sweat, and evaporation. However, the oxygen 18O label, a heavy isotope of oxygen, is lost in both water and carbon dioxide because the oxygen in the CO2 you breathe out comes from your body water pool, which equilibrates with the bicarbonate pool.
Okay, so 18O leaves faster.
Exactly. The 18O gets washed out faster because it leaves via two routes, water and CO2, while 2H leaves mainly via one route, water. By meticulously measuring the difference in the rate of loss of these two labels from body fluids like urine or saliva over an extended period, typically 1 to 3 weeks, scientists can accurately estimate total carbon dioxide production.
And from CO2 production, you get energy expenditure.
Yes. From CO2 production, they can calculate oxygen consumption using assumptions about the likely fuel mix, or the RQ, and ultimately your total energy expenditure over that entire period. This technique provides unparalleled insight into real-world energy expenditure, allowing researchers to study metabolism in people living their normal lives, far removed from a laboratory setting.
So to truly understand our energy needs, we need to establish a baseline for what your body burns just to stay alive, right? The fundamental cost.
Precisely. That baseline is called the basal metabolic rate, or BMR. This is the energy expenditure by your body when it's completely at rest but not asleep, under very controlled conditions of thermal neutrality, meaning a comfortable neutral temperature so you're not shivering or sweating. It's typically measured about 12 hours after your last meal, ensuring your digestive processes aren't significantly impacting the measurement. It's your body's minimum operational cost.
What influences someone's BMR? Is it the same for everyone?
Oh no, it varies quite a bit. Your BMR is influenced by several key factors, providing a personalized metabolic fingerprint. First, weight, or more accurately, body composition. A larger body, particularly one with more lean mass, muscle, organs, generally has a greater amount of metabolically active tissue, meaning more cells that are constantly burning energy, which naturally leads to a higher BMR.
So more muscle means higher BMR.
Generally, yes. Second, age plays a significant role. BMR tends to decrease with increasing age, even if your body weight remains constant.
Why is that?
This is largely because, over time, Metabolically active muscle tissue tends to be gradually replaced by metabolically less active adipose tissue, or fat, which burns fewer calories at rest. And third, gender is a factor. Women generally have a significantly lower BMR than men of the same body weight and age.
Due to body composition again.
Primarily, yes. This difference is mainly due to women's bodies typically containing proportionally more adipose tissue relative to muscle mass compared to men on average. Hormonal differences also play a role.
Understanding your BMR is a crucial step then, in understanding your overall energy expenditure and personalizing your nutritional needs.
Absolutely. Your total daily energy expenditure isn't just your BMR. It also depends heavily on the energy required for your physical activity throughout the day, and importantly, the energy cost of synthesizing reserves after a meal that diet-induced thermogenesis we'll discuss next. Knowing these components allows for a remarkably accurate estimation of an individual's specific energy requirement, taking into account their body weight, age, gender, and their level of physical activity. It moves far beyond a generic calorie recommendation to something far more precise and personalized.
Now that we've covered the baseline, your BMR, let's talk about the energy we burn when we're actually moving and engaging with the world. Physical activity.
Right, the variable component.
The energy cost of various physical activities is very usefully expressed as a multiple of your BMR, isn't it?
Yes, that's the standard way. This is known as the physical activity ratio, or PAR, sometimes also called the metabolic equivalent of the task, or MET. It's basically how many times more energy and activity burns compared to just resting BMR.
Can you give some examples to illustrate the range?
Sure. Sedentary activities like sitting at a desk, watching TV, or light computer work use only about 1.1 to 1.2 times your basal metabolic rate. So not much extra energy is expended there.
Barely above resting.
Pretty much. But by stark contrast, vigorous exertion, such as climbing stairs rapidly, cross-country walking uphill with a load, or intense bursts of exercise like running or swimming, can use anywhere from 6 to 8 times your BMR, or even higher for elite athletes during peak effort.
Yeah, that's a huge difference.
It powerfully demonstrates the incredible range of energy expenditure based on activity intensity, and why an active lifestyle dramatically changes your caloric needs compared to a sedentary one.
So how do you account for a whole day's worth of different activities?
To get a full picture of someone's average daily energy expenditure from activity, we calculate their overall physical activity level, or PAL. This is essentially the weighted average of the PAR for all the different activities someone performs throughout a 24-hour day. You multiply the PAR of each activity by the time spent on it, sum them all up, and then divide by 24 hours.
So it gives an average activity multiplier for the whole day.
Exactly. It gives us a single number that reflects the average intensity and duration of their daily movement and overall energy demands from physical activity, which can then be multiplied by the BMR to estimate total daily energy expenditure.
Here's a fascinating aspect of energy balance that often surprises people and is a crucial, often overlooked, component of your daily energy expenditure.
Ah, yes, the thermic effect of food.
There is a considerable increase in your metabolic rate after you eat a meal. This phenomenon is scientifically called diet-induced thermogenesis, or DIT. It's not just about the calories in the food you consume, but the calories your body burns processing that food. Is that right?
That's exactly right. Diet-induced thermogenesis isn't a single, simple thing. It has a few distinct components. A small part of this increased metabolic rate is the direct energy cost of secreting digestive enzymes into your gut and the active transport of the digestion products from your gut into your bloodstream. That takes some energy.
Okay, the cost of digestion itself.
Yes, but the major part, the truly significant portion, is the substantial energy cost associated with synthesizing your body's energy reserves after the nutrients are absorbed. This includes making glycogen from carbohydrates, building new triacylglycerol fat from fats and excess carbohydrates or protein, and synthesizing new proteins from amino acids.
So storing the energy costs energy.
It does. These synthesis pathways are energy intensive. This is a crucial, often overlooked aspect of energy expenditure, demonstrating that
We've broadly discussed undernutrition, but it's crucial to understand its specific and devastating manifestations. Let's start with marasmus.
Okay, marasmus represents severe energy deficiency.
It's a state of profound and extreme emaciation, and it can occur in both adults and children, affecting vulnerable groups across all populations, not just in specific regions. At its core, it's the outcome of prolonged negative energy balance. Simply, not consuming enough calories over an extended period to meet the body's fundamental energy needs. starvation, essentially.
Yes, chronic energy deficit. The progression of marasmus is quite grim and systematically destructive. Initially, the body's fat reserves are completely exhausted as it desperately tries to find energy to sustain basic life functions. Fat goes first. This is then followed by severe muscle wastage as the body turns to its own protein stores for fuel. Muscle protein is broken down.
Cannibalizing muscle.
Exactly. As the condition worsens, there's a critical loss of protein from vital internal organs like the heart, liver, and kidneys, which severely compromises their function and can ultimately become life-threatening. It's a systemic depletion of the body's own tissues.
In this desperate state of extreme energy deprivation, the body employs a last-ditch survival strategy for fuel, doesn't it?
Yeah.
Using those amino acids from broken down protein.
Yes, amino acids liberated from the catabolism, the breakdown of its own tissue proteins, are repurposed. They are used both as a direct source of metabolic fuel for immediate energy needs and, crucially, as substrates for gluconeogenesis.
Making glucose. Why is that so important?
Gluconeogenesis is the process where the liver makes new glucose from non-carbohydrate sources, like amino acids. This is primarily to maintain a continuous glucose supply for the brain and red blood cells, which rely almost exclusively on glucose for energy and are absolutely critical for survival, even during starvation.
So the body sacrifices muscle to feed the brain.
In essence, yes. But the systemic impact of marasmus extends far beyond just weight loss and muscle wasting. The drastically reduced protein synthesis directly leads to a profoundly impaired immune response, making individuals far more susceptible to recurrent infections and increasing their severity. Infection then further compounds the problem by increasing metabolic demand.
A vicious cycle.
Absolutely. Furthermore, there's a significant impairment of cell proliferation in the intestinal mucosa. The gut lining deteriorates, resulting in a reduced surface area within the gut. This reduction in surface area then further compromises the absorption of any nutrients that are available, creating another vicious cycle of malabsorption and worsening undernutrition. The body literally becomes less able to help itself absorb what little food might be available.
In contrast to marasmus, let's explore kwashiorkor. You mentioned this is a specific and distinct form of severe undernutrition.
Yes, primarily affecting children and historically seen mainly in developing countries. It has very distinct clinical features that clearly differentiate it from the simple wasting of marasmus.
What are those distinguishing features?
The defining characteristic of kwashiorkor is the pervasive presence of edema.
Edema. Fluid retention.
Yes, profound fluid retention, particularly noticeable in the limbs and face, giving affected children a deceptively puffy appearance despite their underlying malnutrition. This edema is strongly associated with a markedly decreased concentration of plasma proteins, specifically albumin, which normally helps maintain fluid balance between the blood and tissues. Low albumin allows fluid to leak out into the tissues.
Okay, edema is key. What else?
Another striking and very visible feature is fatty infiltration of the liver, leading to a noticeable and sometimes painful enlargement of the organ called hepatomegaly.
Now, historically, there was a significant misconception about kwashiorkor, wasn't there? About it being just protein deficiency.
Yes, for a long time it was widely believed to be caused solely by a lack of protein in the diet, while energy intake was considered more or less adequate. This led to the simplistic view of it being a protein deficiency disease, distinct from the energy deficiency of marasmus.
But that view has changed.
It has. A deeper, more modern analysis of the diets and clinical presentation of affected children has shown that this isn't necessarily the sole or even primary cause. In fact, children with kwashiorkor are typically less stunted in their growth compared to those with marasmus, suggesting they might have had slightly better overall nutrition previously. And surprisingly, the edema often begins to improve relatively early in treatment, even when the child is still receiving a relatively low-protein diet, as long as overall care and infection control improve.
That really challenged the protein-only idea.
It profoundly challenged the original protein-only hypothesis. The modern understanding is far more nuanced and complex. Kwashiorkor is now understood to be very commonly precipitated by an acute infection, such as measles or severe gastroenteritis, superimposed on a general chronic food deficiency and often a pre-existing micronutrient deficiency.
So infection is a trigger.
A very common trigger. There's likely a significant deficiency of key antioxidant nutrients such as zinc, copper, carotene, and vitamins C and E in these children. When an infection strikes, the body's immune response, particularly the respiratory burst by stimulated immune cells like macrophages, produces a surge of highly reactive oxygen and halogen-free radicals as part of its action against pathogens.
The body's own defense mechanism.
Yes, but this added overwhelming oxidant stress, combined with the pre-existing antioxidant deficiency, is believed to be the critical trigger that ignites the full development of kwashiorkor, leading to the severe edema, skin changes, and liver dysfunction.
Wow. So the immune response itself contributes to the problem.
It seems so in the context of severe underlying malnutrition and antioxidant deficiency. It raises an important and almost tragic question. How does the body's essential immune response, when not properly supported by adequate nutrition, actually contribute to a further decline in health rather than just combating the infection? It's a cruel irony of severe malnutrition.
Let's look at a third and distinct form of undernutrition, one that is frequently seen in a clinical setting, cachexia.
Yes, cachexia is associated with chronic disease.
This condition is commonly observed in patients with advanced cancer, HIV infection AIDS, chronic heart failure, kidney disease, and a range of other chronic severe inflammatory diseases.
It's fundamentally distinct from simple starvation or marasmus, isn't it?
It is. Understanding that difference is critical for patient care. Physically, patients with cachexia do show all the outward signs of marasmus, extreme weight loss, severe muscle wasting, profound emaciation. However, there is a crucial underlying difference. There is considerably more loss of body protein, particularly muscle protein, than what occurs in simple starvation or typical marasmus.
More protein loss, why? In simple starvation, protein synthesis is reduced, but protein catabolism isn't necessarily dramatically accelerated initially. The body tries to conserve protein. In cachexia, however, the continuous secretion of inflammatory cytokines, signaling molecules like TNF-alpha, IL-1, IL-6, released by the immune system or the tumor itself in response to the chronic infection or cancer, increases the catabolism of tissue protein. This happens via a specific and energy-intensive pathway called the ATP-dependent ubiquitin proteasome pathway, which essentially marks cellular proteins for destruction. This accelerated protein breakdown significantly increases overall energy expenditure, even at rest.
So they're breaking down protein faster and burning more energy.
Exactly. This constant cytokine-driven protein breakdown and increased energy expenditure lead to a distinct metabolic state. Cachexic patients are hypermetabolic.
Hypermetabolic, burning more calories than expected.
Yes. They have a considerably increased basal metabolic rate, BMR, compared to healthy individuals or even those with marasmus, who are often hypometabolic initially. Their bodies are literally burning through energy and protein at an accelerated, inefficient rate, even when they are at complete rest. It's a systemic, destructive metabolic overdrive.
And there are other contributing factors to this devastating metabolic state, making it a truly complex syndrome.
Oh yes, several other factors contribute. Many tumors, for instance, exhibit altered metabolism. They often metabolize glucose anaerobically, meaning without sufficient oxygen, even when oxygen is available, the Warburg effect. This produces lactate, which then travels to the liver to be used for gluconeogenesis, the creation of new glucose, which might feed back to the tumor. This Corti cycle is incredibly energy-consuming for the host, with a net cost of 6 ATP for each mole of glucose cycled.
A futile cycle draining energy.
Precisely. It drains the body's energy without producing a net gain for the host, essentially fueling the tumor at the host's expense. Furthermore, there's often increased stimulation of mitochondrial uncoupling proteins by those circulating cytokines.
Uncoupling proteins, what do they do?
These uncoupling proteins essentially cause the mitochondria, the powerhouses of the cell, to leak protons, generating heat instead of efficiently producing ATP, the usable energy currency. This leads to thermogenesis, increased heat production, and a further increase in the oxidation of metabolic fuels without producing useful energy for the body. It wastes fuel as heat.
More inefficiency.
Yes. And finally, there's sometimes a phenomenon called futile cycling of lipids. Certain factors, including some secreted by tumors, can activate hormone-sensitive lipase in adipose tissue, causing fatty acids to be liberated at an accelerated, uncontrolled rate. These fatty acids are then taken up by the liver and subjected to ATP-expensive reesterification back into triacylglycerols, which are then exported as very low-density lipoprotein, or VLDL. This continuous breakdown and resynthesis of fats, without a net functional gain for the host, represents represents another significant energy drain on the body.
It really highlights the incredibly complex and destructive metabolic shifts that occur in chronic illnesses like cancer and AIDS, going far beyond simple calorie deficits or lack of appetite.
Absolutely. It's a systemic metabolic derangement driven by inflammation and disease processes.
So after dissecting the role of proteins as the body's fundamental building blocks and seeing what happens in deficiency states, how do we actually determine a person's protein nutritional state in a more precise way?
The gold standard for this, particularly in research settings, is by measuring what's called nitrogen balance.
Nitrogen balance.
Yeah.
Tracking nitrogen in and out.
Exactly. It involves precisely measuring the dietary intake and the output of nitrogenous compounds from the body. Protein is the major dietary source of nitrogen, making up about 16% of most proteins by weight. So by measuring the total nitrogen intake, usually from food records or controlled diets, you get a very good estimate of protein intake. The conversion is straightforward. Milligrams of nitrogen multiplied by 6.25 gives you milligrams of protein.
Okay, that's the intake. What about the output? Where does nitrogen leave the body?
The output of nitrogen from the body occurs mainly in urea, the primary waste product of amino acid metabolism, and smaller quantities of other nitrogen-containing compounds like creatinine and uric acid, all excreted in urine. Significant amounts are also lost in undigested protein in feces. This includes not just dietary protein, but also shed intestinal mucosal cells and digestive enzymes, which are proteins themselves. And smaller but measurable amounts are lost in sweat, shed skin, hair, and nails.
So you have to measure all those outputs.
For precise research, yes. By tracking these inputs and outputs, we get a clear picture of whether the body is gaining, losing, or maintaining its protein content.
And the difference between that intake and output of nitrogen is what defines nitrogen balance, giving us 3 key states of protein metabolism.
Correct. First, in a healthy adult who is maintaining their body mass, we ideally see nitrogen equilibrium, where nitrogen intake equals nitrogen output over time. This means there's no net change in the total body protein content.
Equilibrium. Makes sense. What else?
Second, we have positive nitrogen balance. This is typically seen in situations where new tissue is being built. A growing child, a pregnant woman, or an individual recovering from significant protein loss due to illness, trauma, or surgery. In these cases, the excretion of nitrogenous compounds is less than the dietary intake, meaning there's a net retention of nitrogen in the body as protein, allowing for growth, repair, or tissue rebuilding.
Gaining protein. And the third state?
Third, negative nitrogen balance occurs in response to major trauma, severe infection, or burns, or if the intake of protein or overall energy is simply inadequate to meet requirements. Here, there is a net loss of protein nitrogen from the body, indicating that protein breakdown, catabolism, is exceeding protein synthesis, leading to a reduction in total body protein and muscle wasting.
Okay, positive, negative, and equilibrium. Now, it's important to counter a common misconception here, especially given the popularity of high-protein diets these days. Simply consuming a very high intake of protein does not automatically lead to a continuous state of positive nitrogen balance and ever-growing muscles in a healthy adult, does it?
No, it doesn't work quite like that. While a high protein intake does indeed stimulate and increase the rate of protein synthesis, it simultaneously increases the rate of protein catabolism, particularly the oxidation of amino acids for energy. So in a healthy adult who isn't undergoing resistance training stimulus for growth, the body adapts, and you generally maintain nitrogen equilibrium, albeit at a higher rate of protein turnover.
A faster turnover, but still balanced. Like having a faster conveyor belt in a factory, but the amount of product going in still equals the amount going out, just moving faster.
That's a good analogy. It's a higher flux, but not necessarily net accumulation beyond replacing what's lost.
And this increased rate of protein turnover involving both synthesis and catabolism, that has an energy cost, right?
Yes, it's energetically expensive. Both protein synthesis and the pathways for breaking down amino acids, like the urea cycle, are ATP dependent, meaning they require significant energy. This is precisely why you observe an increased diet-induced thermogenesis, that metabolic rate increase after a meal in people who consume a high-protein diet compared to lower protein meals. Their body is working harder just to process and turn over that excess protein, even if they aren't building more net muscle from it.
So given this constant turnover, even in equilibrium, what's the fundamental reason we have a continuous need for dietary protein? Why can't the body just recycle everything perfectly?
That's a great question. Even in a healthy adult who isn't actively growing, there's a continual obligatory catabolism or breakdown of tissue proteins as part of normal maintenance and repair. While many of the amino acids released from this breakdown can be reutilized efficiently for new protein synthesis, a significant portion is irreversibly oxidized for energy or used for gluconeogenesis, especially during the fasting state overnight to maintain blood glucose. This nitrogen is then excreted primarily as urea. This constant, albeit relatively small, loss of amino acids that aren't fully recycled creates the continuous minimum daily need for protein from the diet to replace those losses and maintain tissue function.
So how much do we actually need based on those nitrogen balance studies?
Nitrogen balance studies, which are the basis for our understanding of protein requirements, have provided specific figures. The average minimum daily requirement to achieve nitrogen equilibrium in healthy adults is estimated to be approximately 0.66 grams of high-quality protein per kilogram of body weight per day. To allow for individual variation in needs and protein quality within a population and ensure almost everyone meets their needs, a recommended reference intake, like the RDA in the US, is typically set higher, often around 0.8 to 0.83 grams per kilogram of body weight per day.
Okay, so for an average adult, say 70 kilograms, that's around 56, 58 grams per day.
Roughly, yes. That works out to approximately 55-60 grams per day, or only about 8 to 10% of total energy intake in a typical mixed diet. It's often less than people think is needed just for maintenance.
However, when we look at average protein intakes in developed countries, the reality is quite different, isn't it?
It really is. Average intakes are often in the range of 80 to 100 grams per day, or even higher, which translates to about 14 to 16% or more of total energy intake.
So on average, many individuals in developed countries consume significantly more protein than the minimum requirement needed to simply maintain nitrogen balance and replace obligatory losses.
That seems to be the case. It highlights a common pattern of consumption well above the basic physiological need, perhaps driven by cultural factors, dietary trends, or marketing emphasizing high protein.
While healthy adults have that baseline requirement, certain states obviously increase needs. Growing children, for instance.
Yes, growing children naturally have a proportionally greater protein requirement per kilogram of body weight than adults. This is because they need to be in a state of consistent positive nitrogen balance to support the synthesis and accumulation of new body protein associated with their rapid growth and development.
But how much extra do they need just for growth?
Interestingly, even for them, the actual incremental need for new protein deposition for growth is relatively small compared to the much larger requirement for continuous protein turnover, the constant breakdown and resynthesis that occurs daily just for maintenance. Turnover dwarfs the growth requirement.
Okay, now what about athletes? This is where we challenge a very popular and widely held belief.
Ah, yes, the protein needs of athletes and bodybuilders.
Is it true they need massive amounts of protein?
Based on current rigorous research and nitrogen balance studies in athletes, there is little compelling evidence that most athletes, even those training intensely for strength or endurance, require excessively large amounts of protein far beyond what a normal, well-balanced, energy-sufficient diet already provides.
So the protein powders and shakes might be overkill for many.
For many, potentially yes. Simply consuming more calories overall from a typical balanced diet, where protein makes up maybe 12-15% of the energy, will generally supply ample protein in absolute grams to support the increased muscle protein synthesis stimulated by training. The main requirement for these highly active individuals isn't necessarily a drastically higher percentage of protein, but rather a significantly increased total energy intake to fuel their training and permit that increased protein synthesis and recovery. You need enough energy overall to build muscle.
So energy sufficiency is key. That's a surprising fact for many who associate fitness with huge protein consumption.
It often is. While needs are slightly elevated compared to sedentary individuals, maybe 1.2 to 1.70 kilo a day, depending on the sport and goal, the very high intakes often promoted, over 2 kilo a day, lack strong scientific support for additional benefit in most cases, assuming adequate energy intake.
Beyond the needs for growth and athleticism, certain severe physiological states significantly impact protein metabolism and needs, often increasing loss dramatically. You mentioned trauma earlier.
Yes. One such profound metabolic reaction occurs in response to major trauma. Think severe burns, multiple fractures, major surgery, or critical illness like sepsis. This leads to a notable and often rapid increase in the net catabolism or breakdown of tissue proteins throughout the body, much more than simple fasting.
What triggers this massive protein breakdown?
This increased protein breakdown is triggered by several powerful factors acting together. It's a direct response to the surge in inflammatory cytokines, those signaling molecules like TNF-alpha and interleukins and stress hormones like glucocorticoids, cortisol, which are part of the body's acute stress and inflammatory response. Additionally, there's an excessive utilization of specific amino acids like threonine, cysteine, and glutamine in the rapid synthesis of acute phase proteins by the liver, which are critical components of the body's inflammatory and immune response. The body prioritizes making these over maintaining muscle.
So the body sacrifices its own tissue to fight the stress.
In a way, yes. As a result of this intense catabolic drive, a person can lose as much as 6 to 7% of their total body protein, mostly from muscle, over a mere 10-day period following severe trauma. This leads to significant muscle and organ wasting and weakness.
And another significant factor contributing to protein loss, often overlooked, is prolonged bed rest or immobilization, even without major trauma.
Absolutely. This leads to considerable protein loss, primarily due to disused muscle atrophy, simply because the muscles aren't being stimulated by activity and weight bearing. While protein catabolism might also increase slightly due to low-grade inflammation, sometimes associated with illness requiring bed rest, the main issue is that without the stimulus of exercise, that lost protein isn't adequately replaced, even with good nutrition. The muscles simply waste away from lack of use.
But the body can recover from this, right? Rebuild the lost protein.
Yes, thankfully. The protein lost during these periods of trauma, illness, or prolonged immobility is meticulously replaced during convalescence, the recovery period, assuming the underlying condition resolves and nutritional support is adequate. This recovery phase is characterized by a strong positive nitrogen balance as the body actively rebuilds its tissues. And again, similar to athletes recovering or growing, a normal, well-rounded, energy-sufficient diet is typically quite adequate to permit this replacement protein synthesis without necessarily requiring extreme protein supplementation beyond meeting the slightly elevated needs during recovery.
So providing enough overall energy and standard protein levels is usually sufficient for rebuilding.
Generally, yes. The body, once the catabolic stress is removed and adequate energy and building blocks are supplied, is highly efficient at rebuilding what was lost, supported by rehabilitation and activity.
Finally, to truly master our understanding of protein, it's crucial to grasp that the requirement isn't just for protein quantity, it's fundamentally about the specific amino acids, the building blocks themselves.
Absolutely. This is about protein quality.
Not all proteins are nutritionally equivalent because they have vastly different amino acid compositions. The body's true requirement is for a precise balance of amino acids in the correct proportions to replace losses and synthesize its diverse tissue proteins, from enzymes to muscle fibers.
Precisely. To clarify, amino acids are categorized into distinct groups based on whether the body can make them or not. First, there are the 9 essential or indispensable amino acids: histidine, isoleucine, leucine, glycine, methionine, phenylalanine, threonine, tryptophan, and valine.
Essential meaning?
Meaning they are absolutely vital because your body cannot synthesize them at all or not in sufficient amounts to meet needs. They must be obtained entirely through your diet. Critically, if just one of these essential amino acids is lacking or inadequate in your diet relative to its requirement, overall protein synthesis will be limited like a factory line stopping because one specific part is missing. Nitrogen balance cannot be maintained regardless of how much total protein you consume.
The limiting amino acid concept.
Exactly. It determines the overall usability of the dietary protein.
Okay, 9 essential. What else?
Then you have 2 conditionally essential amino acids, cysteine and tyrosine.
Conditionally essential.
Yes. While your body can synthesize these, it can only do so from specific essential amino acid precursors. Cysteine is synthesized from methionine, and tyrosine is synthesized from phenylalanine. Therefore, your dietary intakes of cysteine and tyrosine directly affect your requirements for their essential precursors. If you don't get enough cysteine or tyrosine from your diet, you'll need to consume more methionine or phenylalanine, respectively, to allow the body to make them. They become essential if the precursor is limited or if synthesis pathways are impaired.
Okay, that makes sense. So that covers 11. What about the rest?
The remaining 11 amino acids found in proteins are generally considered non-essential or dispensable under normal conditions.
Meaning the body can make them.
Yes, these can generally be synthesized by your body from other molecules as long as there is enough total protein, providing nitrogen and energy in the diet. If one of these is omitted from the diet, nitrogen balance can potentially still be maintained because the body can simply make it itself using common metabolic intermediates. However, within this group, only 3 amino acids, alanine, aspartate, and glutamate, are considered truly dispensable because they are readily synthesized by simple transamination of common metabolic intermediates like pyruvate, oxaloacetate, and alpha-ketoglutarate, which are always available from glucose or other amino acid metabolism.
So some non-essential are easier to make than others.
You could say that. And here's the final nuance. For some of the other non-essential amino acids like glutamine, arginine, glycine, proline, serine, the body's synthesis capacity may actually be outstripped by its requirement under certain specific circumstances, such as during rapid growth, severe illness, major trauma, or periods of high metabolic stress. In these situations, these amino acids can become conditionally essential, meaning dietary intake becomes crucial to meet the heightened demand because synthesis alone can't keep up.
Fascinating. This entire categorization of amino acids really raises an important question. How does a deep understanding of these amino acid categories, essential, conditionally essential, non-essential, specifically inform dietary choices?
It's fundamental, especially for individuals with unique needs, specific health conditions like liver or kidney disease affecting synthesis, or certain lifestyles such as vegetarian or vegan diets, where combining different plant protein sources is crucial to ensure all essential amino acids are adequately supplied. It's far more intricate and strategic than simply counting total grams of protein. Quality and balance matter immensely. #tag outro.
So what does this all mean? We've journeyed through the incredible molecular landscape of digestion and absorption, understanding in hopefully quite intricate detail how the body meticulously breaks down and utilizes everything you consume. From the first bite to the final cellular uptake, it's really a testament to biological precision, isn't it? An unseen symphony happening within you every moment.
It truly is. If we connect this to the bigger picture, it profoundly underscores the interconnectedness of diet and health. From the tiniest enzymes breaking down a sugar molecule to the complex metabolic pathways managing energy balance and the intricate regulation of nutrient absorption, every single step is critical and interconnected. We've seen how delicate this balance truly is, whether it's avoiding common deficiencies like iron or vitamin D, understanding complex conditions like cachexia or kwashiorkor, or optimizing athletic performance and recovery. Your body is a master chemist, but it needs the right ingredients and conditions to perform optimally.
You now have a deeper, perhaps even master-level understanding of what it truly means to fuel your body. The intricate dance of macronutrients and micronutrients, the specific transporters, the hormonal controls, and the surprising ways your system extracts every essential bit of nourishment. This isn't just about calories in, calories out, or eat your veggies. It's about appreciating a symphony of biological processes happening in stunning detail that underpins your very existence and well-being.
Absolutely. And perhaps this raises an important question for you, the listener, to ponder. Knowing the incredible precision and the inherent vulnerability of these sophisticated biological systems, how might you approach your own nutrition differently from this moment forward? Not just for today's energy or tomorrow's workout, but really thinking about it for a lifetime of optimal health and longevity. Consider the intricate details, the interactions between nutrients, the importance of bioavailability, and the profound impact of every single choice you make regarding what you put into your body.
A lot to think about. Thank you for joining us on this deep dive. We hope this exploration has given you a fresh perspective and perhaps even sparked a new curiosity about the incredible machine that is your body.